Chapter 12, the chromosomal basis of inheritance. You will be looking at all of these you must knows, but again, you want to be watching the whole video and making sure you're getting all these pieces so you understand the whole picture of it. So let's begin. So chapter 12. So when Gregor Mendel was doing his experiments on pea plants, it wasn't really known what it was that actually transferred the information from parent to offspring. But now we know that that is a chromosome. And so what we're going to do is we're going to take Mendel's laws and we're going to look at the chromosome theory of inheritance, which we've gone over quite a bit so far already. But let's just walk through the entire thing so we can see it here all at once. Now, I know when you see this picture, it looks a little overwhelming, but your understanding of this picture is so essential because this is basically combining everything from Unit 5 so far. So let's first take a look at where are we? So we're looking at 3 generations, the P parental generation, the filial 1 generation, and the filial 2. First, we're starting with pure breeding round and pure breeding green. Remember, pure breed means homozygous. And we're looking at dihybrids. So we're looking at 2 different traits. We're looking at the seed color and the seed shape. Now, when we're talking about a dihybrid, we're talking about 2 sets of homologous chromosomes. So let's look here in this diploid cell. Any cell from the round seed, yellow round seed, you would see homozygous. So here's our Y and our Y, and here's our R and our R. So we have, right, 2 sets of chromosomes that have the Y allele, and both of those chromosomes carry the Y gene. And then the second homologous pair carry the R, right? So these genes, the seed color and the seed shape, are on different chromosomes, right? Different locus on different chromosomes. When this seed is making its zygotes, it can put only capital R and capital Y into its gametes. And that's because that's the only thing they have. So these are the only gametes it can make. If you went through meiosis and saw the result, all of the gametes would look like this. Same thing here, because basically all of the chromosomes for R are little r, and all the chromosomes for Y are little y. The only gametes you can make are little y, little r. Okay, so these are our gametes. So when we're doing Punnett squares, this is what we're drawing. This is the gamete, and we're trying to see, okay, if this gamete mixed with this gamete, what would the offspring be? And that's what you get inside the little boxes of your Punnett square, is the results. And there's many different types. So this R and this R, this Y and this Y, depending on what the gametes are of the parents. Okay, now if you took this gamete and this gamete, now notice here, they're being fertilized. What do you get as a result? Our F1 is going to be a hybrid. It's going to be heterozygous for both of the genes. And that's because this parent gave a capital R for this homologous pair. And then for the other one from dad, we have a little r, right? This one gave a little r. So this homologous pair for the R allele is heterozygous. And then Y, mom gave capital Y, dad gave little y. And so our genotype is capital Y, capital R, little r. So that's what we're talking about when we're talking about this. What this means is what you see here. And the same thing for this one. All the offspring must be this genotype, because the only thing the dad can give is capital R. The only thing the mom can give, or vice versa, is little r. Okay. Now, the F1 generation. Let's say these offspring are trying to self-fertilize. So we're going to cross a heterozygous dihybrid with another dihybrid. Well, first, we need to see what kind of gametes they can make. So we're going to take this one through meiosis, and notice that our homologous chromosomes will pair up and duplicate, right? So that you start off with 2, so you have your capital R, little r, and then they're going to make a copy, right? So now you have a homologous pair. But now we're paying attention to 2 different genes, so we're looking at 2 sets of homologous chromosomes. So we have our heterozygous and our heterozygous. So we have, let's just follow capital R, little r, duplicate, right before. Then anaphase, meiosis 1, they're going to separate. And we call this the law of segregation. So the 2 alleles for each gene are separating into 2 separate cells. So this allele separates to the left, and this allele separates to the right. That's called the law of segregation. And then you get from the first divide, now you have your homologous pairs have divided. And then now the second division, meiosis 2, you're going to have the sister chromatids divide. And now you're making your gametes here. And notice that we can make these options. Or remember that if we go back to this step, you can even align your homologous pairs differently. So you could have the dads on the left and the moms on the right for the R gene, for the R chromosome. and for the Y chromosome, mom on left, dad on right. And so this is called the law of independent assortment, because if you have these homologous chromosomes, they're going to separate, they're going to basically line up independently of the one before it. So they're not, if this dad is on the left and this mom's on the right, that's not going to affect the next, how it lines up at all. They're going to assort independently. And so because of that, because as they segregate through meiosis one, the homologous pairs into separate cells, you actually can get different types of gametes because of the law of independent assortment. And so based on this, these plants going through meiosis, or these parents going through meiosis, any of these, now these are gametes. If they combine, you can create your F2 generation. And so if you did this many, many, many times and you crossed these like thousands of times, you would end up with this ratio 9:3 to 3:1. Okay, so this is an F1 cross fertilization. So this right here is our self cross, right? A dihybrid self cross. Our ratio is 9:3 to 3:1, and that's something you have to memorize. So the F2 generation of a dihybrid self cross is 9:3 to 3:1. Okay, so this is basically putting everything together. Very important that you know this. So now we're going to explore a little bit beyond Mendelian genetics, and we're going to look at non-Mendelian genetics. What's the difference between Mendelian and non-Mendelian? Well, Mendelian genetics is a simple system. We're looking basically at pea plants, and they're genetically simple because most traits are controlled by a single gene. So, for example, the seed shape for pea plants are controlled by just one locus on the chromosome. There's not multiple loci that are determining that gene or that trait. And each gene only has two alleles, and one is completely dominant. So, for example, seed shape, there's either wrinkled or round. There's not like a bunch of other alleles. For example, blood types, right? There are multiple alleles for blood types. You could have the A allele or the B allele or the I allele, but Mendel's plants were simple. But other types of inheritance aren't usually that simple. So if you think of in humans, we're a little bit more complex, right? If you think, for example, for skin color, right? It's not just one or the other, like it is with flowers, where you have white and purple, Mendel's flowers, Mendel's pea plants, which either create white or purple offspring, right? So and we're talking about genetics for most other genes, we're going to talk about different type of genetics. Mendel just kind of paves the way for further research. So one example is incomplete dominance, and this is where if you have two alleles for one gene, so let's say we're talking about flower color, one allele is partially dominant over the other. So none of them really take over completely, and none of them are like totally dominant. They're kind of a hybrid or in between or a blending appearance. So when you think incomplete, think, okay, the dominant allele isn't fully doing its job completely. The recessive allele is also basically asserting their dominance as well. So, for example, let's say you have homozygous dominant, you're going to be red. Homozygous recessive, you're going to be white. But now, if you're heterozygous, you're not going to be red. You're going to be a blend or a mix of both. And in this case, you're going to be pink. Okay, so you will see sometimes the allele is written differently as R prime for the recessive allele, because it's kind of showing that like white isn't recessive. It's just helping out with the dominance. So it's like kind of contributing to the phenotype as well. Okay, so kind of looking at this, so if you took a true breeding red and a true breeding white and crossed them, you would get heterozygous F1 generations. If you did a Punnett square, you would see that 100% pink or heterozygous for the genotype. If you self-pollinate them, we find that you actually get 25% red, 50% pink, and 25% white, and that's a 1 to 2 to 1 ratio. And this is something you're going to want to remember. When you're looking at incomplete dominance, you're going to see these ratios. Here is an example of a Punnett square if you didn't want to memorize it. And here's some different ways of representing the alleles. You could have the capital R for red and the capital R for white. And then if you use the Punnett square, you would find these genotypes, right? So think about, what would this phenotype be? If you have two capital R red alleles, you would have a red phenotype. A red and a white, now instead of having red be completely dominant, now it's incompletely dominant, and now white is showing up as well, blended, and now you have white for homozygous white. And then let's look at our ratios. So here are our genotypes and phenotypes. So our genotypic ratios will be 25, 1 out of 4 is red, 50%, 2 out of 4 is pink, and 25% is white. And then if we write that as a ratio, it's 1:2. The phenotypic ratio is the same because we still have 25% red, 25% pink, and 25% white. So the same ratios. The next type of non-Mendelian genetics is called codominance. This time, both alleles are expressed, but they're not blended. So both alleles are sharing the dominance. They're both basically fully dominant, but now there's kind of like a mix of the two. So, for example, in cows and bulls, homozygous dominant will be red coat, so that will look like this. Homozygous recessive is white, and now if you have the heterozygous, it's like a speckled, or in this case, we call it the roan coat color. And sometimes we use these alleles just as convention. It's not important for you to memorize this. You'll likely be given this in the problem if you get this codominance problem, but just something to think about. Because it's codominant, we don't really write all the time the recessive alleles as lowercase because it's not really recessive, right? It's still contributing to the phenotype, the recessive allele. So you can write it as a different letter, just kind of a different way of writing it. Another example of codominance would be blood types. So blood types is actually an example of multiple alleles and codominance because in this case, you actually have 3 different alleles for what would show up on the chromosome. So you could have an IA allele, an IB, or a little I. Both IA and IB are dominant, and then the little I is recessive. So now you have codominance, they're both. And they're both able to show up in the phenotype, IA and IB. Now, it's important that we know a little bit about blood type because when someone is receiving a blood transfusion, it's important that you're matched correctly. And so let's kind of take a look at this chart here. We went over this in class, but just kind of review. So here is a red blood cell, and on the surface of it, there are different proteins that can be found on the surface. So in this case, the shape of the protein, we call them type A proteins. Or you could have type B proteins on the surface, or you could have a mix, A and B. If you have no proteins, you're type O. And so let's say you have type A blood, your body is going to make something called an antibody. And the antibodies are basically part of your immune system, and they help fight off foreign invaders. But antibodies are specific. They can only bind to certain proteins found on the surface of red blood cells. So if you have type A blood, you're going to have B antibodies in your blood plasma. So your blood plasma is like your blood, the actual blood cells, and then other like antibodies or other parts of your immune system, the fluid outside of your blood cells. Now, if you have B blood, you're going to have A antibodies. If you have type AB blood, you have no antibodies because if you had these antibodies, we'll learn in a second, they would be able to bind to the proteins on the surface. And if an A antibody binds to an A protein, it will cause the blood to clot or coagulate. Basically, it will kind of form, like it won't allow the blood to go through a blood vessel, and that could cause pain, and it could lead to major complications. But in this case, type O, we have both A and B antibodies. Now, when we're doing blood types and we're drawing out genotypes and phenotypes, we have the genotype for type A. If this is your blood type, you could be homozygous for A or heterozygous, IA dominant and recessive. Type B, here are your genotypes, type AB and type O. And we call type AB the universal recipient and the type O universal donor. Another thing to note that we don't really learn from Mendel is that most genes are pleiotropic. What this means is that if you have one gene and it has a certain dominant or recessive nature, it actually affects your phenotype more than just that one single trait. So there's wide-ranging effects due to one single gene. So for example, there's one gene for something called achondroplasia or dwarfism. If someone gets that gene or that allele for achondroplasia, it's going to affect so much about the characteristics of that person, not just one element, right? So another example of this would be like cystic fibrosis. So there's just one gene, and if you get the gene or the allele for getting cystic fibrosis, you're going to have non-working ion channels in your epithelial cells. So these are cells that line many of your organs. This will cause thick mucus to build up on your organs, and this will affect your entire body, not just one part of your body. That single gene will have wide-ranging effects. The same way for sickle cell anemia, there's a gene for this, and if you get the allele for sickle cell anemia, then you are going to be creating an abnormal protein called hemoglobin that's found in your red blood cells. That hemoglobin, its job is to hold on and to bind to oxygen and deliver oxygen throughout your blood in your red blood cells. But if you have this allele for sickle cell anemia, you're going to have abnormal hemoglobin, and you won't have as much ability to bind onto oxygen and deliver it to cells. And so this will have wide-ranging effects on your body and basically how much oxygen your cells can receive. Another type of genetics we need to look at is called epistasis. This is where, let's say you're looking at two genes on two different chromosomes. One gene could mask another. So depending on your genotype for one gene, it could actually have an effect on another gene. So in this case, the two genes are not independent of each other. They're actually related and they're connected. So for example, let's look at coat color in mice. There's two genes that control coat color. So the first is a C gene, and the dominant would be having pigments. So the ability to create pigment proteins if you have the capital C allele. If you have the little c allele, you cannot make pigment. So the second gene, it's another spot on a chromosome. There is an allele for more pigment that's dominant, and you'll make black, or less pigment. So think about, in this case, you're just making more or less, and that's going to contribute to the overall color of the coat of the mice. So let's take a look at one possible genotype. If you have a little c, it doesn't matter what you get, what your genotype is for the pigment allele or for the black or brown gene. As long as you get two little c's, the mice's cells are not able to make pigment proteins at all. So this allele, this genotype will mask the B genotype. So it doesn't matter what the B allele is if you have homozygous recessive C alleles. So you would expect a 9 to 3 to 3 to 1 ratio when you cross two hybrids like we see here. But in this case, let's take a look at this special case. So all the homozygous recessives for the C gene are going to be white. And so that homozygous or heterozygous B will not create a different phenotype here or contribute to our other phenotypes. But if you have a working C allele, as long as you have one dominant C, you are able to create pigments. And so you will have that pigment. Let's look at this mouse right here. So this mouse has a one dominant C, so it's able to make some pigment, but it is homozygous recessive for the how much pigment. So because it's homozygous recessive, it's making a little bit less pigment, so it will be brown. In this case, we have one capital C allele, so we're able to make pigments. We're good. And then let's check our B allele. We have the allele for more pigment, and so we are going to be able to make. We're going to have black mice because we have the dominant allele for more pigment in that case here. Okay, so that's epistasis. We can even look at this in labs. So for labs, there are two genes, either the E or the B allele, or E or B gene. So if you have the capital E allele, you're going to be able to make pigment. Little e is no pigment. And then there's a separate gene for how dark the pigment will be, either capital B black or little b brown. So if you have homozygous recessive for the pigment, the E gene, you will not be able to make dark pigment in the fur. But if you have as long as you have one dominant allele, you will be able to make pigment. So in this case, how do we get yellow labs? Well, if you have two little e's, you're not able to make pigment. And then if you have two little b's here, then you are going to have still, you can't make pigment. So you're basically going to have yellow fur. But let's say you have two little e's here and you have a dominant allele here. Well, you still can't make pigment, so it doesn't matter what that is because you have two little e's. But let's say you do have a capital E allele, you're able to make pigment, but you have two little b's, so that's going to code for brown pigment. But let's say you have dominant, so you have at least one capital E for the E allele, and then you have at least one capital B for the black or brown allele, then you'll have a black lab because you can make pigment and you have the dominant allele for this one, so you can make black fur. Okay, so that's how you get different coats in labs. All right. Okay, so next we're going to look at polygenic inheritance. Poly means many, so we're looking at genes that are affected by two, or characteristics that are affected by two or more genes. And these phenotypes are on a continuum. So it's not like in pea plants where you have green or yellow peas. In this case, you have like a continuum. But you'll see in the population there is a frequency because let's take a look here. So let's say you have these parents and these parents, and we're looking at the skin color gene. So in this case, we're assuming, just for the sake of simplicity, that there are 3 separate genes for skin color, and they're all basically contributing to the color of the skin. In this case, we're looking at melanin production. And so for this gene, we have heterozygous, so we have one dominant, one recessive. The second one dominant, one recessive, one dominant, one recessive. Now, the dominant alleles are going to be making the pigment melanin. And so the more dominant alleles you have here, the more melanin is going to be created. And so in this case, if you have these two that are crossing these hybrids, then here's the offspring that you can make. And notice if you have no dominant alleles, so this would be little a, little b, little b, little c, you're not going to be able to produce melanin, and so you will have this skin color. And then if you go all the way to this side, if you're homozygous for all the genes that have to do with skin color, then you'll be able to make more melanin, and so that will change the skin color as well. So you can see that skin color is a mix of many different genes, and those genes depending. So if you look here, right, this one has 2 dominant alleles for this gene, 1 and 1. So that's going to make this skin color, right? Now, this isn't just skin color, but height, weight, eye color, intelligence, behaviors. These are determined by many, many genes. So it's kind of hard to study this because it's harder to know like which chromosomes or which genes on and which ones are having a greater effect on the phenotype. But just keep in mind that not all genes are as simple as green and yellow. Okay, even eye color. So eye color is a polygenic inheritance, and so many genes determine eye color, and that's why there aren't just a few eye colors. There are so many different types because it's like a blend of multiple genes create the full phenotype. And it's also important to note that it's not just our genes that determine our phenotype. Nature also, or nurture, I guess, our environment also controls our phenotype as well. So, for example, these hydrangeas, their color is influenced by soil pH. So, depending on what the soil pH is, it will change the color of its phenotype. For humans, our skin color is influenced by our genetics, but also by our environment. So, exposure to ultraviolet radiation will stimulate cells in our skin called melanocytes, which are cells that produce melanin. And so, depending on how much UV light is received, it's going to determine how much more melanin is produced. And this melanin actually works, these melanin proteins work to actually protect the nuclei of melanocytes from ultraviolet radiation, so acting kind of like as an umbrella, so that the nucleus of those cells are not damaged by more sunlight. So, like when someone is receiving lots of sun, it's going to trigger melanocytes saying, "Hey, try to make more melanin because we need to protect our nuclei against more ultraviolet radiation, which could potentially damage our DNA." So Gregor Mendel was able to use his pea plants to study and basically predict what would happen in phenotypes if certain parents were crossed and predict what the offspring would be. But it wasn't really known how this was happening until the chromosome theory of inheritance, which kind of started to come around in the early 1900s when scientists started understanding DNA better and how DNA is transferred from parent to offspring. And part of this comes from experimental evidence and improved microscopy, so microscopes, and now starting to use animal breeding. We are able to understand genes beyond what Mendel saw. Mendel had no idea what DNA was at the time. So we're kind of mixing everything together now that we have the full picture, but we're going to kind of take a look at some other things that were discovered beyond Mendel. And something we want to look at is Drosophila studies. Drosophila is a scientific word for fruit flies. So we'll look at some research done. So Thomas Hunt Morgan was an embryologist at Columbia, and he was the first to actually connect the idea that, okay, genes are found on chromosomes, and you could connect a specific gene to a specific chromosome. And so he bred fruit flies. And it just kind of was a happy accident that he discovered this. He was working with fruit flies, and fruit flies are prolific. They breed very quickly, 2-week generations, so they create a lot of offspring. They actually are also very simple genetically, so they only have 4 pairs of chromosomes, and the last pair, so basically three pairs, and the last pair is the sex chromosomes, which is similar to humans in that XX is female and XY is male. So Thomas Hunt Morgan, he was in his research lab and he noticed that all of his fruit flies had red eyes. And it's important, this vocab word you're going to want to know. The red eyes, we call it the wild type. Okay, wild type. You want to remember that. Wild type basically means what's normal. It's what's found in the wild. And he found that most of his flies were red eyes. And one day, Morgan actually was looking at his fruit flies and he found one fruit fly that did not have red eyes. It actually had mutated eyes. So their chromosome actually had mutated. And he was able to actually look at the chromosome and see which chromosome had changed between the red eye and the white eye fruit flies. And he was able to connect that gene to a specific chromosome and say, oh, here's where the change is. There's a difference between this chromosome and this chromosome, and was able to kind of locate the location or the locus of that gene and tie it to a specific chromosome. So what he actually figured out was something called sex linkage, and this is where genes are actually found on sex chromosomes. And we're going to take a look at how this is inherited. So let's take a look at his experiment. So Morgan found that if he took a red wild type, so wild type, remember, is the normal, the red eyes, and he crossed it with a white-eyed male, all of the F1 generation is going to be red, which is kind of similar to Mendel's experiments. But when he crossed the F1, the hybrids, he found that the F2, 100% of the females would have red eyes and 50% of the males would have white eyes. But all his females would always have red eyes. So he's kind of looking at a different pattern of inheritance than Mendel had been predicting when he was doing his F1 and F2 generations. So how is this possible? It's something called a sex-linked trait. So let's take a look at what he did. So I know that this slide looks a little bit complex, but we're going to walk through it, and it's important that you're kind of following along carefully so you can understand this. So he did an experiment. He took a wild-type red fly, and he crossed it with, that's a female, here's a female sign, crossed it with a mutant white-eyed male. And he found that in his F1 generation, all of his offspring had red eyes. His F2 generation, he found that 100% of the females, so 2 out of 2 of these are red, and then 50% of the males are white and 50% are red. And what did he conclude? Well, let's take a look over here at this diagram. Well, let's start with the P generation. So if we actually were to look at the chromosomes of this female, we would see 2 X chromosomes because the female here has 2 X. And on the chromosome, we have this allele, and we're going to use the W allele with the plus sign to show that's the wild type. Okay, the wild type is the normal red eye allele. This will make red eye proteins. And then this one, it's homozygous, so we have 2 normal wild type alleles. But let's look at the male. The male on his X chromosome has a mutant allele. So without that plus, this is saying mutant. And then the Y is basically doesn't have that gene on it. So it is not carrying that allele at all. So when this female is making eggs, she can only put the wild type allele in her eggs because that's all she has in her chromosomes. So that F1 will have this from the mother. But then what kind of sperm can the father make? Well, the sperm can either pass down in the sperm cells the mutant allele or the Y allele. And so let's do like a little Punnett square. So if we take the dad's sperm with the mutant allele and the mom's wild type allele, we have a heterozygous. And in this case, we're going to get a red phenotype. So we have that mom's wild type, so the phenotype will also be wild type or red because it's masking the father's mutant allele. But let's look at if we make male. So these are 2 X chromosomes, right? So that's a female child. But let's say if the dad passes down his Y chromosome and the mom passes down her wild type, her normal red allele, you're going to get a normal son because even though that Y chromosome doesn't have the wild type allele, still it's saved by the mother's chromosome, and so you're going to have red eyes. But what happens when this male son creates gametes? Well, this male could either put the Y chromosome in one sperm or the wild type chromosome in another sperm. The eggs here, or this for this female, can put the wild type or the mutant allele on her X chromosomes. So if this egg is fertilized by this sperm, then you'll have normal wild type alleles, homozygous, and you'll have a normal female right here because you have two X chromosomes and they're both wild type. Let's say you have a wild type and a mutant, you will still have a female with a normal red wild type phenotype, red eyes, because even though you have the mutant allele, it's masked by the wild type allele. But this one is now a carrier. So that's important to flag. Now, this son will have red eyes because its mother donated the wild type chromosome and the father donated a sperm or a Y chromosome. And so that will be a male, but it will have normal eyes. But then this one, the dad donated the Y chromosome and the mom donated the mutant allele. And so we don't have any backup. So we only have that mutant allele. So the son will be mutant. It'll have white eyes. And that's why the son are more likely to have the white allele, because if it gets that white or that mutant allele from the mother, it doesn't have the backup from the father to create the wild type red eyes. So these traits that are found on sex chromosomes, the X or the Y chromosomes, are called sex-linked traits. And so in humans, similar to the fruit flies, we have two sex chromosomes. Two X chromosomes develops a female, assigned female at birth, XX, and there's redundancy. So these two chromosomes have the same genes on them. So you have two copies of each gene on your X chromosomes. But if you're a male, assigned a male at birth, then you have an X and a Y, and there's no redundancy. This means all your X genes, there's only one copy of them, not on the Y. And then here's like a picture under a microscope, and you can see the X is much bigger, and the Y is just a little guy, less genes on it. And we've already kind of talked about this, but just a reminder, in your normal body somatic cells, if you're a male, you have 44 autosomal chromosomes. These are the chromosomes that determine everything else in your body except for like your sex organs and some of your sex hormones, which are determined by your sex chromosomes X and Y. And so if you have X and Y male, and sperm cells, you're going to have half of those. These are haploid, 22 autosomes and one sex chromosome. Okay, and so the same for the female. We have 44 autosomes, two sex chromosomes, and our diploid cells. This is our haploid, and this would be a new zygote. So male and female. And this is just a few Punnett squares on how to determine the sex of the offspring based on the parents. And notice here that, let's say the mother has an X chromosome with one gene on it that's dominant, and the mother also has a gene on it that's dominant, then she can only make dominant allele eggs. If the dad has a gene that's recessive and a Y, he can only pass down a recessive sperm and a Y. And so if you do the Punnett square, you can see the offspring. So this baby will have the dominant and a recessive. This is a female because it has two X. And this one will not have this one has the trait because it has one dominant. This boy will also have the trait because it's dominant. This will be the female. It will have the trait because it has one dominant. And this one will also have the trait. If you look over here, you can see for this boy right here, it actually does not have the trait because it has a recessive allele and also a Y chromosome. And so it doesn't have any instructions for building whatever this capital N is supposed to be. Okay, and the same here. If the sperm and the egg both pass down two little n's, so the mom's in this case a carrier, then you will see that it's possible for a female to get the trait or two little n's here because she now has two copies of the recessive allele. So again, we kind of talked about this in past units, but a diploid cell is 2N, has two copies of each chromosome, and the last chromosome X and Y, two copies, either XX or XY. So here would be XY. Now, sex-linked traits, the Y chromosome is really small, so it only has a copy of like a few of the genes, so like right here. But most of these genes up here, they're only found on the X chromosome. Okay, so you would be diploid for a gene here because you have two copies. You have homologous chromosomes here, but not up here. All right, so if the X chromosome has a recessive allele, then it has to be expressed because that's all that's there. So the recessive will show up in the phenotype because there's nothing coming from the father. Okay, we've already kind of gone through this. Now, let's look at the Y chromosome for a second. There is a gene on the Y chromosome called the SRY, the sex determining region. And this is like the master regulator for maleness or like male like sex organs. And so this turns on the gene for the production of male hormones. And this is an example of pleiotropy, where one gene will have a great effect on the whole organism. So this one gene, the SRY gene on the Y chromosome, will determine so many of the traits of the male. And then the X chromosome has a gene called DAX1. This is called anti-testes factor. But if you have the SRY, so if you have the Y chromosome, it inhibits DAX1. And so you don't have this anti-testes factor. Now, there are other traits beyond sex determination on the X chromosome. So, for example, like baldness and color blindness, these genes are also found on the X chromosome. There are genes also for hemophilia or Duchenne muscular dystrophy. And so these are also potential genetic conditions that could come about if you have that gene on your X chromosome. And color blindness. So the human X chromosome, if you take a close look at this, you can see all the genes that are found on the X chromosome. And so as long as you have one copy of that gene or that allele, if you're a male, you will have that trait because your Y chromosome doesn't have that gene on it. So as long as you have one copy, you'll have it. So sex-linked, when you're talking about sex-linked, we're talking about genes that are found on the X chromosome. And there are more than 60 diseases traced to genes on the X chromosome, including color blindness and what we just talked about. Okay, so here is the Y chromosome. So like I just said, there is the testes determining factor, or SRY, and then 30 other genes on the Y chromosome. So if someone had the Y chromosome, then they would have this, which would basically regulate the production of male hormones and also like male sex organs as well. But let's talk about sex-linked traits. So let's look at two normal parents. So we're just going to like create our own gene, for example. So in this case, we have our father, which is going to be determined by this square here, and our mother is going to be a circle. And the mother is a carrier for a sex-linked trait. So the father is homozygous dominant, and the mother is heterozygous. She's carrying a sex-linked trait. But when we write the alleles, we're going to write X because she has two X chromosomes, capital H and lowercase h. And then the dad actually just has one allele for this because it's sex-linked. It's only found on the X chromosome. This gene is not found on the Y chromosome. And so this father is normal because he has the working copy of this gene. But the mom also normal because she's heterozygous. She has one working copy, so she's good. Now, let's say the dad is creating gametes. So the dad can either create dominant or Y gametes. Someone's knocking at my door. The female can create dominant or recessive sex chromosomes, right? In the egg cells here. So when we do a Punnett square, the dad can either donate a dominant X or a Y chromosome in the sperm, or the female can donate a dominant or recessive. And so when you do the Punnett square, let's say dad donates X, mom donates X, you're going to get a female, two X's, and they're both dominant, so you're not going to have that trait. Let's say mom donates X, dad donates Y, you're going to have a male, XY, and it's going to be normal. Someone's still knocking on my door. Okay, so if egg donates X and male donates X, and we have the mom donate the little h, the recessive allele, you're still going to be normal. You're going to be a female, but you have one working copy, so you're fine. But if your mom donates a little h and your dad donates a Y, then you have a male that has that trait because in this case, you have the allele. And then this one, you actually wouldn't have it because you're still recessive here, or you still have a dominant allele here. So how would you know if something is sex-linked or X-linked? You want to look at the X chromosomes. You want to see um, if the males get their X from their mother, it's possible that they could get the recessive allele from their mother and then the Y from their father. And if they have that recessive allele from their mother, if their mom's a carrier, then it's possible that they could have that disease. The trait is never passed from father to the son. Think about it. The father can only pass Y chromosomes to the son, right? If the father has a daughter, then the dad can pass his X chromosome in the sperm to the daughter. But if you are a son, then you got your Y chromosome from your father, and so you can't get X-linked chromosomes from your father. So if you're looking at, we'll look at this in a second, but for example, like baldness or colorblindness, these X-linked traits, they must come from the mother. So it means the mother was a carrier for that trait and gave you her X chromosome that had that different gene on it, that colorblind or that bald gene on it. There are very few Y-linked, but there are some Y-linked traits. There are only 26, around 30 genes on the Y chromosome. And so these would be traits only passed from father to son because only the males have the Y chromosomes and only the father can pass the Y chromosome to the son. But the females can't inherit the trait because the female doesn't have a Y chromosome. The next type of non-Mendelian inheritance pattern is called X inactivation, and this only happens in females, so you have to have two X chromosomes. And in this case, we're looking at females who have heterozygous alleles on their X chromosomes. So we're looking here at a calico cat, and we want to know, okay, why does calico cat have like speckled appearance here, with some fur that is black and some fur that is orange? And that's because when this embryo first began, when the sperm and egg combined, one parent donated the allele for the orange fur and one parent donated the allele for the black fur. And after this zygote has formed, it's now going to divide through mitosis and make a ball of cells, which will continue dividing. Now, after a few cell divisions, some of the cells will actually inactivate one of their X chromosomes. So in this case, we're going to turn off the X chromosome that has the orange fur allele. And so only the black fur is going to be activated in this cell here. And so all the cells here are only going to have the instructions for making black fur. And so wherever you see black fur, that's because the cells that make that fur only have the black allele active. In the same way, this cell, it's divided through mitosis, but it's inactivated its allele for the black fur. And so it only has active orange allele. And so you'll have parts of the fur that are orange. So this is how you can get this speckled appearance because of X inactivation if you're turning off one of the sex chromosomes in females. All right, now we're going to look at linked genes. It's kind of a new topic here. And this is going to be important because this type of inheritance is, again, different than Gregor Mendel's inheritance. So let's take a look at this sperm here and this egg here. And we're looking at 3 different chromosomes. And this is a haploid cell because we have one copy of each chromosome. Now on this chromosome, we have a gene D, just one gene here for the sake of this picture. This chromosome, we have 4 different genes. This chromosome, we have gene E. Now, the genes that are on different chromosomes are not linked, okay? And we call them assorting independently. So we'll look at meiosis in a second, and we'll see that when these chromosomes are going through meiosis, they're going to separate from the other chromosomes during meiosis. We'll look at that in a second. But if you see that there are 2 genes that are close to each other on a chromosome, we call them linked genes. And that's going to be important when we're doing chi-squared because you're going to see different phenotypic and genotypic ratios when you do Punnett squares for linked genes. It'll come up in the chi-square. So we have 1, 2, 3, 4, 5, 6 different genes here that we're looking at. So if we're looking here at this, if this sperm and this egg combine, now you're going to have homologous pairs. So the dad for this chromosome 1 and this chromosome from the mom, you're going to have right here and right here. So your homologous pair is heterozygous, capital D and lowercase d. And you can see that showing up here in the genotype of this child. For the A, you have this chromosome here, and this homologous chromosome is heterozygous. For B, you have this chromosome and this chromosome. But now, notice here that the A, the B, the C, and the F genes are all on the same chromosome, okay? But really, the ones that are close by on the same chromosome, we call linked. So A, B, and C here are linked genes. And we're going to take a look at how that works in a second. Now, each chromosome has hundreds or thousands of genes, but in this case, we're just looking at 4 different genes here, and the ones that are close together are linked. So just like I said, genes located near each other tend to be inherited together in genetic crosses. So unlike in our Mendelian genetics, we actually follow the law of independent assortment, where we say that alleles are going to be separated from each other through meiosis. This is going to happen differently, and we're going to get different probability rules. Okay, so let's take a look at an example here. So here's our mom and here's our dad, and here's a homologous chromosome pair. And we're looking at 2 different genes on the same chromosome that are linked. So here we have our B allele, and this is a wild type. So we don't know what it is for yet, but just kind of follow along here. And this is another allele. It's a VG wild type. And then on the other chromosome, you have 2 mutant alleles. So this is the mom's, and then this is the dad's. And the dad has all mutant alleles here. And these are just normal chromosomes. These aren't sex chromosomes. And so the dad has 2 copies. What are you going to get for the offspring? Well, let's see. The mom can donate this chromosome, and the dad can only donate the same, right? Both mutant alleles here. Or you can have the mom donated this chromosome and the dad donating this chromosome. Okay, so the genes are linked here. They're coming together. They're not on separate chromosomes. Okay, so how does this affect inheritance? So let's look at a case study, an example that was done on fruit flies. So we're looking at Drosophila, and we're looking at, okay, body color and wing size. We want to know, are these genes linked? Are they inherited together? So if you're going to get, for example, the gray body color, does it always have to pair up as a package deal with the wing size, like this normal long wing size? Or are they not linked? Mendelian genetics. So we're looking at linked versus non-linked. Linked means the 2 genes are inherited together. They're a package deal. If you have one, you're going to have the other. So let's do an experiment with a P generation. We're going to take a pure breeding. So we have homozygous dominant for both. And this B+, this is going to be the gray allele. This is the wild type. So this is the normal. The B, just a regular B, is the mutant allele. So that is going to be black. The VG+, that is normal wing size. That's the wild type. The VG is vestigial. Vestigial means like short little wings. So these are short wing size. So here we have homozygous wild type for the B allele. So we have gray appearance. And then vestigial homozygous wild type. So normal wing size. And that's what you see in the phenotype as well. Crossed with homozygous recessive. So both mutant alleles for the color, body color, and both mutant alleles for the vestigial wing size. So we have a pure breeding here. We're going to do a cross here between the P generation, and we're going to get our first hybrid. So notice here that you could get here a heterozygous. So you have the wild type allele and then the mutant allele, so heterozygous, but you're still going to have the wild type phenotype. So you're going to have the gray, the normal phenotype. And then this one will receive 2 little alleles, and this one will have 2 little alleles here. So this is the offspring of these parents, and we're going to take this offspring and we're going to do a test cross. And so again, remember, a test cross is you're going to take your F1, you take your dihybrid, and you're going to cross it with homozygous recessive for both alleles, these 2 alleles. And we want to see, are these alleles linked? So if they are not linked, if they follow Mendelian genetics, what do you expect the phenotypic ratio to be if you do an F1 dihybrid test cross? Take a moment and think, when you do a test cross, what is your expected ratio? A test cross. If you said 1 to 1 to 1 to 1, we did this in class, then you're correct. So let's see if we actually get a 1 to 1 to 1 to 1 ratio when we test these. If we do get that, then the genes are not linked. If we do not get a 1 to 1 to 1 to 1, then that means the genes are linked. Okay, if we don't get 1 to 1 to 1 to 1, then the genes are not. Then the genes are linked. Okay, so again, we are crossing here a heterozygous. So we have B+ and B, that's the allele here. And then the vestigial plus the wild type with the mutant, this is heterozygous. So this is a hybrid, and we're going to cross it with homozygous recessive for the B allele and the vestigial allele, the wing allele. Okay, so this is what we saw just now. And now when you cross your heterozygous, your hybrid, with your homozygous recessive for both genes, then here's what you're going to get. So you are going to see that here's the father. So the father can only donate little b and little vg. So no, it has to donate the mutant alleles here. So the sperm can only be this genotype, okay? But the eggs, the mom could make, if you do your foil, right, the mom could make this, or this. So if you foil this out like we did in class. So if this egg and this sperm combine, you are going to have B plus and little b, so wild type and mutant, and you'll have heterozygous, so you'll have normal appearance, normal gray appearance. If you have vestigial, so this would be the wild type, and then the mutant, you have heterozygous here, but you still have that dominant allele, the wild type allele. So you're going to have the normal wing size. Here you have little b and little b, so you're going to have black appearance. So both alleles are mutant. You're homozygous recessive. And the same thing here, and you can follow along for these two as well. So this gray, we have B plus and B combined to get our heterozygote, and then 2 VG, so these two are mutants, and that would be homozygous recessive here, so vestigial traits, etc. So you would expect if the genes were on different chromosomes that you would get a 1:1:1:1 ratio, because that's what you usually get when you do a dihybrid test cross. So dihybrid test cross is when you get two genes are heterozygous crossed with two genes that are homozygous recessive. But the results show from this data that we get 965 to 944 to 206 to 185. So this many of this phenotype, this many of this phenotype, this many of this phenotype, and this many of this phenotype, which comes out to a 1:1 to 0:0. So basically these ones, there's like so few of them that we mostly have these if you work out the ratios. So this does not follow Mendelian genetics. And so this would show that the genes are actually linked. So where you're going to see this come up is in chi-squared problems. If the ratios don't come out to 1:1:1:1, then you will actually find that you're going to be rejecting the null hypothesis. And you'll say, this does not follow Mendelian genetics. This is a linked gene for if you get rejecting the null hypothesis. And we'll do some practice in class as well. Okay, finally, we're going to talk about chromosomal abnormalities in humans. So if we look at this karyotype here, what happens if there are changes in this karyotype? So the average human has 46 chromosomes or 23 pairs, but what would be the reason why you might have more or less chromosomes? It has to do with an issue in meiosis called non-disjunction. So let's take a look here at a normal diploid cell, so 2N, so we have homologous pairs, and they've already made a copy of their DNA. So we have two sister chromatids, and they're lining up on the metaphase plate, and they're independently assorting. So dad's on the left, mom's on the right, they're independently assorting. And remember our steps of meiosis. So first we're going to do meiosis 1. We're going to separate our homologous pairs, and we're going to divide those into two new cells. And eventually, these three are going to go up into this cell, and then these three chromosomes will go down to this cell. So that was meiosis 1. And then meiosis 2, these sister chromatids will split. So you will divide those, and then you will get 4 daughter cells. Right here, it's just showing you 2, but there are more. There are 2 of these and 2 of these. And we would have half the number of chromosomes, and we have 3 of each because each of these split in half. Okay? And so this started with 6 chromosomes, 1, 2, 3, 4, 5, 6, and now you end with 3 haploid. That's normal meiosis. So what happens in non-disjunction? Well, let's start over. So let's say you have your diploid cell, and then let's say during meiosis, the chromosomes, these tetrads, these homologous pairs, don't separate correctly. So you're going to actually, when they line up on the metaphase plate, only some of the chromosomes will go into one of the daughter cells in meiosis 1. So here you have 1, 2, 3, 4 chromosomes going in this one, and only these 2 going in this cell. And then meiosis 2, they'll divide again. The sister chromatids will separate. And now you have 2 chromosomes in this one and 4 chromosomes down here. And so they did not separate correctly. And so now you have too few or too many chromosomes in your egg cells or your sperm cells. And so then when the chromosomes combine, so like let's say if these are eggs and they combine with sperm, you're going to have 1 extra or 1 less chromosome. So this is 1 less and 1 more. And then here's another example. So this is non-disjunction. So let's say we have this cell and we have, when they're lining up on the metaphase plate, these two homologous chromosomes stick together and they don't separate from each other. Then you'll have 1, 2, 3 of these chromosomes going into this cell, and then just this one chromosome going here. And then this chromosome during anaphase will separate, this one will separate, and this one will separate, and then you'll end up with 3 chromosomes here and 3 chromosomes here, whereas you were supposed to start with 4, right? That's the haploid number, and then 2 in the gametes here. This one only got 1 of these chromosomes, and so when it split during anaphase, now it has 1 less than it should have. It's supposed to have 2 because the original diploid cell had 4, it's supposed to have 2 here, but that didn't work. Or if you see here, non-disjunction can happen during meiosis 2, where this one we separate the sister chromatids, or we separate the homologous chromosomes here, and they go into separate, and then the sister chromatids don't separate in this part here. And then now you have 3 copies of the chromosome here, 1 and then 2 and 2. Okay, so there are different types of non-disjunctions, either during meiosis 1 or meiosis 2. And basically what you need to remember is that there is a problem during anaphase with the separation of homologous chromosomes or sister chromatids. Now, what will happen is the sperm and the egg now have more or less chromosomes, and so when they combine, the zygote has the wrong chromosome number. And we call this aneuploidy. So this type of this word basically means the wrong number of chromosomes. Now, trisomy means you have 3 copies of a chromosome. So that's if you had 1 extra. And then monosomy is if you only have 1 copy of the chromosome. So for example here, you have 1, 2, 3, 4 instead of the 3 that you're supposed to have. And so if these sperm and egg combine, you're going to have an extra of 1 of these because the mom is actually going to have 1, 2, 3 of this one, or two of the same one here, and the dad has this one, or monosomy. So this happens a lot in humans when zygotes are being formed, but what happens is the embryo will usually be miscarried spontaneously. So the pregnancy won't be able to be completed because changes in the chromosomes are too disastrous. Because if you have too many chromosomes or too less chromosomes, too many chromosomes means you're making too much protein for those genes, and that will be a biochemical imbalance. So you're not going to be able to basically develop properly. Too few chromosomes means you're not going to be able to create the right proteins to develop properly. Now, certain conditions are tolerated. So if you have an extra chromosome or 1 less, and it doesn't really upset the balance as much, you can survive. And if you have an extra or less chromosome, aneuploidy, then we call this a syndrome because you have a set of symptoms. So it's not just one thing that is the effect of this. There's many things, and this is a syndrome. So for example, Down syndrome, this is trisomy, so 3 chromosomes on number 21. 1 out of 830 children are born in the US with Down syndrome. It's the smallest human chromosome, so it has the least number of genes on it, but it has severe effects. And the frequency of Down syndrome correlates with the age of the mother. So if you look here at this chart, if the mother is under 30, then 1 in 1,000 births, there's a 1 in 1,000 chance of having Down syndrome. But if the mother has the pregnancy at age 49, 1 in 12 chance. So as the mother ages, you can see that the incidence of Down syndrome out of 1,000 births increases rapidly, like kind of after age 35. Okay, but there can be genetic screening before the embryo continues to develop. So amniocentesis in the second trimester of the pregnancy, you can take some of this amniotic fluid and you can test out some of the fetal cells, and you can actually make a karyotype so you can see all the chromosomes, and you can actually know if your developing embryo is going to have trisomy or too many or less chromosomes. Okay, there are also types of genetic conditions where you have extra or fewer sex chromosomes. And human development, we're more tolerant of the wrong numbers of sex chromosomes. So it's more likely that the embryo will be miscarried if there is a wrong number of chromosomes in like chromosome 5 or chromosome 6, right? But the sex chromosomes, it's okay if there's extra or less. but it produces different conditions. So you could have two X's and a Y. This will be Klinefelter syndrome, XXX, or XYY, or XO. So this would be just an X chromosome. And this would happen, again, during non-disjunction. So, for example, if the female, and when she's going through meiosis to make egg cells, she could accidentally put two, not accidentally, but because of non-disjunction, because the chromosomes don't separate, like I showed you, you could have an egg cell with two X chromosomes, and then one egg cell with no sex chromosomes. And so the sperm, depending on what the sperm donates, you could have, if the mom donates two X's, you could have XXX because the dad donates the X, or the mom's two X's with the dad's Y, XXY, etc. So this is how you can get it if one of the sperm or egg donates more. Construction. Interesting. Okay, Klinefelter syndrome. So this would be an XXY male, 1 out of every 2,000 live births. The male has sex organs, but they're sterile, so they can't produce offspring. The extra X is inactivated, so it's still just XY, but there can be some breast enlargement, and also this male will be typically tall and just have normal intelligence. So again, you can see XX, two X's, and a Y. Or you can actually have 1, 2, 3, 4 X's and a Y. That could also happen. Jacob syndrome, so X and two Ys, 1 in 1,000 men, an extra Y chromosome. You're a little bit taller than average. You're a little bit more active. You might be more active. Slight learning disabilities, delayed emotional immaturity, but average intelligence and average sexual development. So this would be a Jacob syndrome male here. Or trisomy X, so 3 X chromosomes, 1 out of every 1,000 live births, and produces healthy females. Why is that? Well, it's because some of the X chromosomes are inactivated. So because of that, it's not really going to affect the offspring at all. So if you see here, here's your zygote, and you have 1 X and 2 X's. One of the X's is going to be inactivated anyway, so it's not going to be a big deal. Okay, Turner syndrome is monosomy X or X0, so only 1 X chromosome. This is the statistics of it. Varied degree of effects, so there's not just like one typical outcome here, but typical intelligence, webbed neck, short stature, and immature sterile females, so cannot produce offspring. Okay, so the last thing we want to look at here is what else could happen to chromosomes? Well, if you look closely at the chromosomes could actually have deleted segments, and so that could affect the chromosomes as well. Inversion, so the genes here, the pieces of DNA can be swapped around and switched. So as you see here, a change, there's a reversal of the segment. So BCD becomes DCB, so the genes kind of switch around. Duplication, so you could add a piece of DNA. Remember, chromosomes are made of DNA. Or translocation, so one segment of a chromosome can move to another part of the chromosome. And so this will also have effects on the appearance and the characteristics of a living organism as well. But these are some other ways that the chromosomes can change. Okay, lastly, mitochondrial DNA inheritance. So when a female and male are producing offspring, we have our oocyte, or this is another fancy word for the egg cell, and here's our sperm. And when the sperm is fertilizing the egg, basically just the DNA from the sperm is going to go into the egg, and all the other organelles of the sperm cell are not going to be passed down to this new zygote, the combination of the sperm and egg. So what that means is the zygote gets all of its mitochondria from its mother. Now, why is this important? Well, remember the endosymbiotic theory, the theory that perhaps mitochondria way long ago was it's actually its own prokaryotic cell. And actually it was engulfed by another cell. And over time, over many years, that prokaryotic cell had a symbiotic relationship with the one that engulfed it. And so it ended up becoming an organelle that can basically help the now eukaryotic cell metabolize or break down food like the mitochondria does to generate ATP. Now, prokaryotic cells, their DNA is circular. And so you will see actually in mitochondria, the mitochondria has circular DNA. Mitochondria can divide on itself through something called binary fission. So basically the mitochondria divides by itself. but it has DNA inside. So all of the DNA that's inside of mitochondria was inherited from mother. So for example, let's say sperm and egg combine and you make a baby girl, then the DNA in the nucleus of the zygote comes from the mom and the dad, right? They share. But what about outside here in the mitochondria? The mitochondrial DNA only came from the mother because the mother passes down her mitochondria to the children. And so all children will have their mother's mitochondrial DNA because the mother's cytoplasm basically is what the zygote will be inheriting, not the father's cytoplasm, which means the mitochondria will not be passed down to the child from the father.